Welcome to today's Learn at Lunchtime program from the State Museum of Pennsylvania. I am Beth Erickson. The topic today is Leading the Way with eDNA, and with us is Luanne Steffy, an aquatic ecologist with the Susquehanna River Basin Commission. Luanne has worked at the River Basin Commission on stream, river, and lake monitoring for more than 20 years. Since 2019, she has been working with environmental DNA sampling as a screening tool for invasive fish species. Welcome and thank you for presenting the program today.
Sure thing. I'm happy to be here. Thanks for having me. I appreciate the opportunity.
Sure. So tell us a bit about yourself and what you do as an aquatic ecologist.
Well, like Beth said, I've been with the commission for a little over 20 years. I am not native here. I grew up in the South in various places, but I've been in Pennsylvania for longer than anywhere else. I did my undergraduate work at Geneva College in Western Pennsylvania and my master's degree at Drexel University in Philadelphia. And I ended up here in the middle of the state since about 2004. In addition to the eDNA work that I do, which is just kind of a small part of my job, I'm currently also working on harmful algal bloom research, dam removal, impacts of dam removals on small streams, long-term data sets, and some work with EPA and their national aquatic resource surveys. So I'm happy to say that after 20 plus years, I still get excited at this time of year. It's almost field season. I can get out and be in a stream again, and I'm pretty excited about that coming up pretty soon here.
That's great. Well, let's get to your presentation to learn more. While you get set up, let me remind the audience that this webinar is being recorded, and by participating in this webinar, you are consenting to the recording, retention, and use of any statements recorded as part of this webinar. If you have a comment, please place it in the chat, and we'll get to respond to that as soon as possible. Please submit questions about the presentation into the Q&A, and we'll respond after the presentation is complete. Luanne, when you are ready, please begin.
All right. Thanks again, Beth. I appreciate the opportunity to be here. I'm going to talk to you a little bit about eDNA today and hope to give you some information maybe that's new to you and to explain some of the cool stuff we do with it here at the Commission. Like Beth said, I do work for the Susquehanna River Basin Commission, and in case you are not familiar with where that is, the Susquehanna River crosses 3 states. It begins in New York State up by Cooperstown, which is in sort of the top right corner of your screen where my cursor is. It flows down through central Pennsylvania and then dumps into the Chesapeake Bay and Havre de Grace, Maryland. So it's a large basin, about 27,500 square miles. So we have a lot of ground to cover. Major tributaries you may have heard of are the West Branch Susquehanna out this way, the Chemung River up here in New York State, and the Juniata River here kind of in the southwest corner. A couple key characteristics. Susquehanna is the largest freshwater input to the Chesapeake Bay. About 3/4 of the freshwater to the bay come from Susquehanna River, and we're about 60% forested, which is pretty good for a basin this size. So that's where we're working at. That's kind of our study area. We have a lot of ground to cover, so eDNA helps us do that more efficiently in some cases. So what is eDNA? I'm sure you've heard of the DNA. You might not remember what it stands for from your high school science class. It's deoxyribonucleic acid. We'll just say DNA here. And if you watched any TV crime shows, you know that it's a way that you can be identified. Each person and species has unique DNA that you can be uniquely identified by. So just like you could be identified at the scene of a crime by your DNA, we can use DNA to identify fish species in our samples. The E in this eDNA just means environmental. In our case, it's mostly water, but there is eDNA work in air and in soil as well. So the kind of eDNA work that we do is called targeted eDNA sampling, which means we're looking for specific species, in our case, fish. So geneticists, which I am not one, people who are much smarter than me and work in a genetics lab, have developed markers that match the DNA of a specific species, and they're called primers or markers. And if the DNA of a certain species you're looking for is in the water sample, the primer will attach to it, and the DNA will, what we call, amplify and replicate itself, and it'll be a positive detection on the machine for that sample. The process is called qPCR, which you may again remember from your high school biology class. It means quantitative polymerase chain reaction. Just a fancy way to say that DNA replicates a lot of times, so it becomes visible on the machines. It's quick and reliable and pretty inexpensive. You may have heard qPCR in the context of COVID testing. Same general process. If the swab that you took from yourself had COVID DNA matched with a COVID primer, it would replicate itself and it would show up as a positive. And if not, it'd be negative. So you may have heard of qPCR sort of in that context. So I'm going to talk just briefly about how we collect the samples. This is sort of our role on the front end. Traditionally, you see my coworker here in the top right collecting a water sample. It's pretty low tech. It's just you wear gloves and you have a clean, sterile sample bottle, and you label it and you fill it with water, and then you filter the water and freeze the filter. Like I said, pretty low tech. And the bottom right corner, you'll see sort of our filtering setup. We have some manifolds where we can filter 8 sites at a time, but you can maybe imagine that it's lots of changing gloves and keeping things dirty, clean and dirty, and keeping samples from being contaminated and making sure you're pouring the right sample into the right cup. And there's lots of risk of contamination. So far, we've been pretty good about that, but there is a higher chance with this method. We still use this method some, but it's kind of the traditional method that we use mostly in the river when we can't wade. A newer method of eDNA collection and filtering is this pretty new on the market eDNA filter backpack. You can see here one of our interns last summer wearing it. At the bottom, the base of this kit is just a filter pump. It has a computer that can track location, amount of water filtered. It'll tell you if your filter is being clogged up, and it kind of keeps track of all the important metrics and measurements that you need when you filter. On the end of this yellow pole is this little filter. The thing right here, kind of where my cursor is. The clear tube goes down into the water, and the filter sucks the water up from the creek through the tube. The filter is the first thing that the water hits, and everything else past this top of this filter is waste. So that's just comes to the filter pump and then gets sent out the output tube back into the creek. So you can probably already tell this is much less contamination, much less chance for contamination. This filter is not touched by anyone's hands. Like I said, it's all self-contained, it's self-preserving. There's no need to freeze anything. So we have been using this more and more in recent years for streams that we can wade like this. It takes about 2.5 minutes to filter 2 liters of water. So it's a little more time in the stream and then just a scoop of water, but it certainly saves time on the back end and also saves the chance of contamination and messing up the filters. But both these filters, the filter that you would kind of see here at the end of this filter cup here and the bigger filter from the backpack, they both go to a genetics lab for processing. And that's not something that we do ourselves. So I'm going to talk about 2 main applications that we use for eDNA in Susquehanna. The first one is to monitor the spread of invasive fish species. This just means fish species that are not native to the Susquehanna and they shouldn't be here, and they are potentially harmful to native fishes and native ecosystems. And the second is to track the success of reintroductions of native species like the American eel, which I'll talk about second. So the first application here with the invasive fish species, the two fish you see here, northern snakehead and blue catfish, are not native to the Susquehanna. The snakehead is an Asian fish that was introduced a couple decades ago. It's a voracious predator. It'll eat basically anything. It can live out of water for a small amount of time. It's hard to kill, and from what I hear, it's really good eating and really good fishing. So they are being perhaps intentionally introduced in some places for fishing. The blue catfish is a very large, can be a very large catfish. It's native to the Ohio drainage, and it was introduced in Virginia somewhere, and it's made its way up into Chesapeake Bay and into Susquehanna, the mouth of Susquehanna. So if you know anything about the Susquehanna, you might know that we have 4 very large hydropower dams. I have them here on kind of in red rectangles here on the map, kind of show where they are generally. Again, this is Harrisburg. Chesapeake Bay is down here at the bottom. Conowingo Dam is the first dam. It's the picture you also see here on the left. That's in the state of Maryland, and the other 3 are in Pennsylvania. So the dams historically have stopped these fish, the blue catfish and the snakehead, from moving upriver because there's a big dam, and so they can't go upstream. But inevitably, like I said, they're sometimes intentionally, sometimes unintentionally through unintentional fish lift introductions, can get over the dam or spread upriver. And we use eDNA to kind of track the progress of how that's going, how fast it's going, where they're invading, so that we can kind of tailor our message to anglers and the public on what to do if they catch one, and please don't release it back into the water and that kind of thing. We do have a whole story map and dashboard on this work. It's been going on since 2019. If you want all the details, I encourage you to take a look at that. I think Beth might put the link in the chat later, and this QR code, it will also take you there. But I'm going to give just a really brief example of some of our maps that show how we track, in this case, the northern snakehead up through the Susquehanna. So just to orient yourself here, this is a 2019. The red triangles are kind of where the dams are. So this is our first year of sampling. We sampled pretty heavily below Conowingo Dam here, the first dam. We weren't sure, what we'd find, how widespread things would be. So all the orange dots are where we had eDNA hits for northern snakehead. And this is a tributary that comes in below the dam. So there's snakeheads the whole way up here because there's nothing blocking their movement. And also there's one hit down kind of in the river on the edge. If you looked at 2021, 2 years later, you'll see we didn't sample as many places below the dam in subsequent years, since we knew they were already there. So you don't see a lot of hits below the dam, but you do see some hits starting above the dam. So in 2021, we saw snakeheads in Conowingo Pond, which is the pond here between Conowingo Dam and Holwood Dam. And we also had snakehead in one of the tribs that flows into Conowingo Pond. And that was 2021. We also had a hit out here in a pond in a park in Lancaster County. Now we had kind of heard reports that there may be snakehead in there, and there was indeed. And then in 2024, you'll see, sadly, a lot of pink dots, and you'll see snakehead DNA was found above all the dams, between the 2 lower ones, the middle ones, the whole way up above York Haven, which is the most upstream dam. And also, if you know Lake Redmond in York County, and this is a small pond in Mechanicsburg. And this is actually in that same trib that was full of them before, but this is above a dam, even in this trib up here. If you know Octoraro Creek, they are above the reservoir there too. So you can see how they are spreading, like I said, either through intentional or unintentional means. They're spreading, and so we use this information to work with our partners and agencies to try to spread the message more and target outreach and education to anglers to not spread them and not put them back in the river. I'm going to show an example of what kind of data we get back. So I said we send these filters to the lab, and what the lab does is some extraction of the DNA off the filters, and then they take 8 replicates off of each filter of the DNA that they extracted, and they'll run it against the primer that we're looking for. So they'll run it against snakehead first, and they'll tell us how many of those 8 replicates were positive for snakehead. And they'll also tell us the next column is mean cycle threshold time. So how quickly the DNA showed up on their machine, basically. And if you hit around 40 or 41, they kind of stop it and say there's no DNA. So the lower the number, the faster the DNA shows up, which kind of gives you an idea that the more fish may be in that system. And also the third thing that we get from the lab is copies of gene per liter. So it's, again, another relative way to look at differences between sites. So we can use this information to kind of make educated understanding of where there might be more or fewer of these species. So like this first row here had 2 replicates positive out of 8 for snakehead and no replicates positive for blue cat. And then this site down here at the bottom, no snakeheads, but 4 out of 8 replicates for blue catfish and a pretty high copies per liter and a pretty low, pretty fast cycle time. So this is the kind of information that we get, and I'll kind of wrap back around to this at the end on kind of next steps for our research. All right, I'm going to change gears from invasive fish species to native fish species. So we also use DNA, eDNA, to track the success of American eel reintroduction. So the same dams that keep invasive species from moving upstream also stop native migratory fishes to move upstream, which is not good. And so you may not know, but American eels are migratory fish, and they spawn in the Atlantic Ocean in a place called the Sargasso Sea. And then once they're little baby eels, like you see in this bucket, which you'll see in a minute, moving around, they're called elvers, and they make their way from the ocean up into fresh water, like Chesapeake Bay, and then up to the Susquehanna, and then they kind of get stopped because of the dams. So this is on the right, there's a picture of an eel ladder, an eel ramp. So there's a small dam here on Octoraro Creek. Eels can come the whole way up here from the bay, and then they get stopped right here. So we have this little eel ramp. It's like a slide, basically, that has water trickling down, and the eels are attracted to flowing water. So they slide their way up this ladder and drop into a large tank, which is checked very often. And the eels are put into a big tank and moved upstream above the dams by the tens of thousands. And here's a little picture of the little elvers that we catch on those eel ramps. So you saw how big the basin is. How do we know where how eels are doing, where they're going, how well they're thriving in certain populations? Are they succeeding? Are they establishing populations? Are they growing? How do we know any of this information? So the dots all show where eels have been stocked. The larger dots are larger numbers of eels. The sites are the same, but the numbers of eels is probably a couple years old. But you see that we've dumped 700,000 as of 3 years ago around Harrisburg here. This is sort of near Edders, above 3 Mile Island. And this is right near Conduit Creek mouth here near Harrisburg. And the other ones, big stockings in Sunbury, Lock Haven, up in Pine Creek, and a couple other small ones. So we use eDNA to kind of sample all this white space to see where the eels have gone. Here's a few pictures of some eels that we caught working. This is probably the biggest one I've ever seen, or close to it. We caught last year. This is about as big as they get before they migrate back out to the ocean. Okay, so let me back up 1 second here. You see this little dot right here by Harrisburg, number 12. That is Beaver Creek. And if you know this area at all, Beaver Creek is a tributary to Swatara Creek. If you know where like Nye Road is in Harrisburg, Beaver Creek is around there. So as part of a project we have here, about 10,000 little elvers were stocked in Beaver Creek in 2016. And we stocked them in the late summer, and there was a couple of high flow events after that. But we never caught any eels with traditional electrofishing methods between 2017 and 2024. We never could find them. We didn't know if they were washed out by the high flows, or they just didn't establish well, or if some sort of water quality event was an issue. So in 2023, we sampled for eDNA on 8 sites on Mainstem Beaver Creek, and what you know it, all 8 had eel hits. So the eels are there, they're established in the whole watershed, all the way up to the headwaters, which is sort of above 81, way up at the top where you can almost hop across it. There's eels the whole way up. So even after we were unsuccessful, and so were other agencies or other consulting firms, they all did look for eels with electrofishing, didn't find a one, but they are there. So we can use eDNA in that way to track success of stockings. The second sort of case study here with the eels. So New York State is about 25% of our basins in New York State. And if you recall from the map of stocking sites, not many are in New York State. There's 2 little ones that were up here near Norwich and near Oneonta in about 2018 or 19. Maybe only about 5,000 or 6,000 eels, elvers at each one. But you know, eels can move great distances, and we've stocked in the West Branch and North Branch, and so you know, we just didn't know how many eels were in New York part of the basin. So we partnered with some universities and did some work of our own too. And the little green dots are places we've actually caught eels, physical specimens. The larger green dots are where we have eDNA positive hits. So some of them are co-located and some of them are not. And then the brownish dots are places where we sampled for eDNA but did not have hits yet. So we did not sample very much in the Chemung Basin. There may be some other places with eels there, but we have not sampled as much there as of yet. This is a couple examples of some of the streams up in New York State. There's my coworker using the backpack there at Snake Creek, I believe. So a couple of next steps I wanted to mention. Like I said, when I showed that data sheet a couple of minutes ago, we just sort of get a presence absence and then like a relative abundance. So we I can't say at this site there's 20 eels, or at this site there's 5 blue catfish. There's no way to kind of quantify it to that level. So we can say this location probably has more because the sample amplified faster, or this site probably has more because eight replicates were positive compared to this site over here, which only had 3 that are positive. But the next step of what we want to do is using that third variable that we get from the lab, which is copies of the gene per liter. And so we're trying to start using traditional methods of electrofishing, especially for eels, where they're a little bit more common than the snakehead or the blue catfish would be. And so at the same time or the same season, certainly take an eDNA sample and a traditional fish sample, and then make some quantitative relationships between density of the fish, density of eels. So maybe 150 eels per meter square fished, and that you get a copies of gene per liter of 400. And if you get enough of those relationships, you can sort of have a regression equation, and then going forward, you can just use copies of the gene per liter to give you an idea of how many fish might be there. And it's probably species specific, and it's going to take a while to get this done, but we're starting to make some of those sort of more quantitative relationships with some of this data. And I just want to mention, sort of at the end here, you may have heard the term metabarcoding in some of the eDNA things you may have heard of. It's not the kind of eDNA we do. You might recall I said at the beginning we do targeted eDNA monitoring. So we're looking for one specific species, or in some cases, 2 specific species. So they'll run the same sample for the snakehead, and they'll run the sample again with 8 replicates more for blue cats. So metabarcoding is when you would send a whole water sample to a lab. And if you're interested in, say, algae, you can say, tell me all the algae that are in here. Or if you're interested in fish, you can say, tell me all the fish that are in here. You're not interested in like one species, yes or no. You just want to know what they find in the water. And so if you remember what I said about you have to have a parameter that matches up with the species to detect it. If the species that's in the water is not in your gene bank in the computer system that they're looking for the primer for, it won't show up. So this method, this sort of metabarcoding science is pretty new. There's still a long way to go, but it is certainly in the future, certainly they'll be able to tell you all the fish that are in a water sample just by a water sample, not by actually catching the fish. In my experience so far with this sort of work, I've done a little bit for a couple other projects, and say for instance, you might have 5 darter species in your creek. The machine might spit out darters as opposed to greenside darter, banded darter, shield darter, that kind of thing. You might just get darter. So the effectiveness of this sort of metabarcoding is only as good as the genes that are available. So you're going to miss some species, especially rare species. Common ones are probably going to be pretty easy to get. But this is kind of the next wave of eDNA research, but it's sort of a long way off, relatively a long way off. And before I take questions, I would like to acknowledge the Commission for funding this research since 2019, for believing in this novel sort of monitoring and the importance of it, and the importance of the information we're giving to this region as pretty much the only ones who are doing this until recently. I said we send our samples to a lab. All the lab work is done by the U.S. Fish and Wildlife Service Northeast Fisheries Center for Genetics in Lamar, Pennsylvania. and appreciate all the work they've done for us there. They actually had to create the marker for us for blue catfish. There was none, so they did the work to create that marker as well for us back in 2019. And Aaron Henning is my co-lead for the lower Susquehanna invasive DNA work. He's also he runs our eel program, so all the eel videos and pictures you saw were probably his. So I would be very happy to answer any questions that you have today, or if you want to email me later, I'm happy to take questions that way as well. Thank you so much.
Luanne, thank you so much for that presentation. Let's get to some questions. And we did put some links in the chat as well for you to contact Luanne after the presentation. So first question, about how many samples does the SRBC collect each year?
Well, we do about 60 to 70 in the lower Susquehanna for snakehead and blue catfish. And then we have been doing about 25 to 30 eel samples around the Susquehanna as well. Later on in the year, as we're doing other work, we will do eDNA kind of as we go around.
And the DNA in the water that's being collected, does that come from remnants of the fish, like scales that have fallen off, poop? What exactly would be leaving that behind?
That's a good question. I meant to mention that earlier, and I thank you for bringing that up. Yeah, it can come from mucus, skin cells, decaying fish carcass that's just sitting in the water that someone threw back, fecal material, waste material, scales, I guess whiskers of a catfish. Pretty much anything of the fish that's in the water can kind of slough off DNA, and it only persists in the water. There's some debate about how long. Depends on how fast the water's flowing and kind of light degradation and things. But the general rule of thumb is like less than a week, a couple days to a week that that will persist in the environment.
Are surrounding states using this technology as well?
Maryland is. They are doing some snakehead work in the Chesapeake Bay and some of the tributaries in Maryland, and I think Virginia as well. New York State is just kind of warming up to the idea recently. I think they were a bit skeptical when we first started, but I know that an invasive species nonprofit in New York is starting to use eDNA this coming up summer, I think, for the first time.
Would you talk a little more about the danger posed by the existence of non-native fish species in the Susquehanna River?
Sure. Well, the blue catfish are certainly voracious predators. They eat about anything. You may have seen a picture online sometime of a blue catfish with a duck halfway sticking out of its belly. They'll eat just about anything, and they outcompete native catfish, which is the channel catfish in the Susquehanna. So it can disrupt food chains that way, as it can outcompete native species. But and that fish is, like I said, it's just in the Ohio Basin, and may have less impacts. Maybe not less impacts than snakeheads would, but snakeheads are like a top predator, and like I said, might have said before, they're voracious. They can live outside of water with barely any oxygen. They like to live in weedy areas, and so they are top predators that can definitely disrupt the food chain, eat a lot of the kind of middle food chain level size fish, wipe that out for other native fish species that would kind of collapse the food chain possibly. So they're kind of inevitable, and maybe in 50 years they'll have kind of adapted to be part of the regular ecosystem food chain. But right now we're kind of unsure how they affect it, so we're trying to keep them out as much best we can.
Is it illegal to introduce non-native fish to the river?
Well, the blue catfish was introduced by a state agency, so I'm not sure how to answer that question. I don't know if it's illegal, specifically. It's certainly frowned upon, and it's hard to trace where they originally came from. There's a couple other invasive species that were probably brought into the Susquehanna by like bait buckets, bait fish brought across watersheds. I think snakeheads, I think they were aquarium fish perhaps, and then got let go, and then they were realized to be, like I said, they're good fighters. So if you're an angler or fisherman, they are fun to fish for, and so they get spread quite a bit. We've heard many accounts of people seeing them being spread.
Well, we'll switch the subject a little bit here. So, is eDNA being used for other invasive organisms?
Is it? You said?
Yes. Yes, sorry.
Oh, that's okay. Yeah, it is for sure. We
Or is there potential to do more with it?
definitely potential. We do have one other small project here at the commission that we used. If you've ever heard of a zebra mussel or a quagga mussel,
Mhm.
we used eDNA to make sure that we had eradicated quagga mussels from a quarry that we're using for water supply or for water augmentation to the river during low flows. So it is very possible if you can have someone, or if there is a primer marker made for that species, it can be used for it. Yes.
You mentioned a creek or pond in Cumberland County above the Dock Street Dam that had a hit for snakehead. Someone wanted to know what creek or pond that was.
Oh, yeah, sure. It's a very small pond kind of near Messiah College. So, University College, I'm not sure. I should ask your husband, Beth, sorry. Um, um, I think it's called Grantham Park.
Oh, yeah, Grantham Pond. Yeah.
Yeah, Grantham Pond.
Okay.
Um, we had a report from Fish and Wildlife Service a couple years ago that there was snakehead. They went out, tried to catch them and like get rid of them out of the pond. I think they might have caught one or two, but they might not have caught all of them because 2 years later we had DNA in there still. Thankfully, there was no DNA in the tributary coming out of that pond or in the Yellow Breeches, which is the next big tributary to it. So they're just in that pond, at least as of last May.
They sure do get around.
They do.
So, whose responsibility is it to monitor the eel fish ladders to disperse the eel to get them to go upstream?
Yeah, so that's a good question. My coworker could answer this more clearly, probably. He's in charge of all the fish passage work, but I will do my best. So the dams on Susquehanna are hydropower dams, and as part of their certification to be dams and to make hydropower, they have to provide fish passage in some capacity. And so the state agencies, us, Fish and Wildlife Service, DEP, Maryland, Fish and Boat, all meet regularly with the consultants for the hydropower dams. And the consultants for those hydropower dams are the ones that monitor very closely the eel ladders and do all the truck and transport of the eels. I should mention too, this is interesting maybe, there's a fish lift at Conowingo, and since all the invasive species hits that we got below the dam in 2019, they started sorting every fish before it goes into the fish lift. And they probably don't catch everything, but they sort all the snakeheads and blue catfish and also flathead catfish, actually. All those species get sorted out before the rest of the fish get lifted over the dam. So that is one management action that has happened since we found all the eDNA in that area right below the dam in 2019.
It sounds like it takes a lot of collaboration between agencies throughout the state and around to keep the river healthy.
Yeah, for sure. It's a lot of coordination.
So, what can the general public do to help with the eDNA program or other programs that the Susquehanna River Basin Commission is working on?
Well, certainly if you are somewhere and you see a sign that says there may be snakeheads here or other invasive species, if you catch a snakehead, please don't return it to the river. Eat it or dispose of it in other ways. Just do not put it back in the river. Education, tell your friends, and just sort of be aware of outreach events like we have sometimes that sort of spread the word about not moving fish between basins and between streams.
Thank you, everyone, for your questions. Luanne, what would you like the audience to remember from today's presentation?
Well, I hope you learned a little something, and I hope that I convinced you that eDNA is a powerful and useful tool, sort of in our toolbox of monitoring techniques. I don't think it's going to replace traditional monitoring methods quite yet. So I think my job is secure as far as that goes, but I think that is certainly up and coming, and I think you'll probably hear more about it in the future. So I hope you got a good background of it today. Thank you.
Thank you again for this presentation. If the audience has further questions about today's topic, you can contact Luanne Steffy directly at the Susquehanna River Basin Commission. Please join us for more Learn at Lunchtime programs. Visit the State Museum of Pennsylvania at statemuseum.org for program information and to sign up.